some of the most pressing issues in the modern immuno-oncology is to identify biomarkers that can predict patients' outcomes, the patients who are treated by immune checkpoint inhibition specifically. This is a very particular problem because currently what we know is somewhat limited. Current biomarkers that we have on the table are powerful in a certain context, but very limited in explaining a large heterogeneity of the outcomes that patients really manifest. So we definitely need biomarkers that can have a bigger predictive capability, but what is most important, that have a personalized predictive capability. We strongly think that our immune system, our host immunity, is somewhat determined by germline genetics, inherited genetics that somewhat modulate our immunity to the point that that immune response may affect how we respond to infections or how we respond to oncological diseases. Not only from the standpoint of therapies, but potentially how our immunity may shape a disease progression and risk, of course. In the context of immuno-oncology, we strongly hypothesize that these germline genetic markers may be very important surrogates for helping us understand which patients would respond to these drugs, which patient will not, and most importantly, which patients will develop potential toxicities from these therapies. We have published some data that have shown that some germline genetic factors, specifically those that impact our susceptibility of developing autoimmune diseases, may be modulators of immunotherapy efficacy, immunotherapy response. Now we are building a new consortium, big consortium of investigators across the world that can pool their resources, clinical data, specimens to tackle this problem more broadly, and developed a large genetic scan involving looking through entire genome for the genetic variants that may associate with these immunotherapy outcomes. In collaboration with more than 50 centers across the world, we are now developing a big project that can really pool close to 20,000 patients together to address what is the role of these germline genetic factors in immuno-oncology and how can they be used from the purpose of predicting the efficacy and toxicity to these therapies. What is also very important is the fact that these germline genetic factors, once identified and validated, will not only serve as potential predictive biomarkers, that at the end they will show us the biological pathway that is important in this process. So we are very optimistic in this, and we strongly suggest that such big consortium work may really identify targets, biological targets that can be used as modulators of the current therapies. We can perhaps, based on the determination of each individual's inherited germline component, devise novel biological treatments that can somewhat complement immunotherapies either before or after treatment, so that patient would be somewhat determined to reduce toxicities that result from this treatment, and also not interrupt the treatment efficacy. These germline genetic markers could be very complementing and reliable alternative to what we currently know.